I'm Daniel Levine, and this is the Bio Report. When Vanderbilt University Medical Center began to consider how big data and precision medicine were going to impact both research and development and clinical care, it struck on the idea of building a large-scale biobank and tying it to its store of millions of electronic health records. That ultimately led to the founding of Nashville Bioscience, a for-profit subsidiary of the medical center. We spoke to Leeland Ekstrom, co-founder and CEO of NashBio, about the company's data collection methods, the significance of the alliance it's developed with leading biopharmaceutical companies, and how it's enabling a transformation of drug development with data. Leeland, thanks for joining us.
Glad to be here today, Daniel. Appreciate the opportunity to speak with your audience.
We're going to talk about genetic and real-world biomedical data, NashBio, and its efforts to make large data sets available to help discover new drugs and accelerate their development. Before we talk about what NashBio is doing, though, I'd like to start with how it came about. NashBio is short for Nashville Biosciences. It takes its name from the city where it was founded. It's a bit unusual that it's a for-profit subsidiary of Vanderbilt University Medical Center. Can you start by explaining how NashBio came to be and how you became a co-founder?
Yeah, it's an interesting story in that regard, and it starts probably about 10 years before I joined the picture. My colleagues at Vanderbilt Medical Center were very forward-thinking in terms of how big data and ultimately precision medicine or personalized medicine were going to impact both the research and development activities and clinical care, and were the ones who came up with a lot of the ideas that we benefit from right now. The idea of building a large-scale biobank and tying it to their electronic health record in a fashion that's optimized for research. All these things came about 2004 or 5 timeframe. So I said about 10 years before I came out of the picture, and it powered a very, very rich research ecosystem at VUMC in that regard. The founding impetus for NashBio was actually market driven. And what I mean by that was that they had received a number of inbound queries from life sciences companies, pharmaceutical biotech, about how to potentially use these data sets. At the time, the UK Biobank was still growing and kind of moving out into the world, and a lot of these techniques were becoming more and more familiar, or more and more essential tools. And so, recognizing the resources that Vanderbilt had accumulated, there was a lot of interest in figuring out a channel to do that. There were some stops and starts in that regard, and particularly around trying to use the sponsored research model for some of these things, which we can spend a long time talking about some of those challenges, but it's not terribly customer centric in that regard. And there can be challenges in contracting when especially you're dealing with 2 large institutions, trying to get things going in a timely or quick fashion, as well as just kind of dedication of focus. And so NashBio was created to address all those problems. I was recruited to VUMC around the summer of 2015 timeframe to help write a business plan. I was part of a large consulting firm at that point, and we were retained to help write that business plan. They kind of take all these different teams together and propose a viable business opportunity that could help VMC make use of these data assets outside of its research enterprise. As we got through the writing of that business plan, I was essentially invited to help build the business, and that's what I've been doing for the past 10 years now. So coming up actually about a month away from my 10th anniversary, and building a lot of those principles developing a lot of those principles we started with in terms of customer orientation and speed and flexibility and nimbleness, but obviously iterating through a bunch of different ideas of what we're actually selling to the market. And I think that's been a lot of the interesting stuff we've done over the time since we first launched the company.
And what's the relationship with Vanderbilt beyond its ownership? Does it continue to provide a source of data? Does it have other roles it takes within the business?
You're right about the ownership stake. We do have a board that manages from a strategic and governance standpoint, and then from a lot of our operations activities, we do operate pretty independently. Vanderbilt is our primary source of data, and that goes back to the kind of the two big domains I mentioned a few moments ago. We leverage a de-identified copy of Vanderbilt's electronic health record, so all of the data that gets collected in the provision of clinical care, the interaction between patients and the various healthcare providers at a place like Vanderbilt. That flows into the main clinical electronic health record, and then goes through a series of transformations to ultimately come out into a common data model that is research ready and then de-identified under the different provisions of the HIPAA statute. And so we have access to both the structured and the unstructured components of Vanderbilt's electronic health record data. Structured is things like labs and medications and visit history and kind of all the easily quantitative or quantifiable parameters. But then also we have access to a number of the unstructured components of that health record as well, things like clinical text and medical imaging and some of the more interesting or the kind of where a lot of the color commentary sits about the healthcare that people receive. We also have access to a large-scale biobank called BioView that Vanderbilt has stood up, is collected from residual biospecimens secondary to clinical care. And what that means in lay language, basically, if someone consents to participate in the biobank, it's only if they have a clinical blood draw somewhere in the health system. We then get access to that residual material. The DNA gets extracted and stored, and then we can go on and genotype or sequence it as demand allows. And so we get both of those feeds coming together. We NashBio, this is where some of the independence comes in. We set off on our own infrastructure. We're an entirely cloud-based organization, and that goes to some of the customer-friendly and flexibility aspects that I highlighted as well. That we found just having ability to build our own infrastructure allows us to move much more quickly and develop in ways that being part of a larger institution.
We produce massive amounts of health data today. Actionable information is another story. What's been the challenge for drug developers, both in terms of accessing data and getting it in a usable and actionable format?
Yeah, it's a good question, and I think that the difference between data and insight is a very key distinction to keep in mind. I often come back to like a resource extraction analogy, mining, if you will, where having large quantities of healthcare data of the types I described really are akin to a mine or maybe raw ore sitting in the ground, so to speak. And so, like keeping in mind how that data is collected and why it's collected. Well, it's collected for clinical communications between physicians and healthcare providers. It's collected for billing purposes in terms of ultimately billing the insurance systems that we have here in the United States. It's collected for quality control measures in terms of the how the hospital is running. So a whole bunch of reasons where the primary uses are not necessarily research uses, and that's why I say think of it as kind of raw ore that has to be extracted and refined in that regard to ultimately make it useful for the kinds of secondary activities that we engage in at NashBio. We have to pull it out of the underlying databases that the health records feed into. It has to be normalized and into what's called a common data model. So in our case, we use something called OMOP, which is an observational data model that's it's quite common in a research setting. Basically, that means mapping it into a data structure that others are going to be familiar with, so you can start to set these data sets, put these data sets together, and then, as I said, de-identified. So really, that's just the next step in processing and kind of building on our resource analogy. Maybe we've moved it from raw ore to refined steel or something in that regard. But then to be useful for the end user, whether in the pharmaceutical space or the diagnostic space, you still have to take it steps further. And so thinking about things like completeness of the data set, being able to track longitudinality, the level of curation that goes into the data set. Those are all steps that we can apply in varying degrees of activity as working with our clients. We are working on kind of standardizing some of those things. But it's important steps to kind of get to useful data that people can use and not having to spend a ton of time cleaning or otherwise munging data to bring it all together. And then once you've done all those steps, that's when you actually start doing the subsequent analyses. So whether you're doing a genetic analysis involving maybe a genome-wide association study or maybe something in the biostatistic space, looking at significant or non-significant treatment patterns in different patient populations. But if you don't do all of those steps, it's really difficult to do a meaningful analysis there. And really, that's one of the key things that NashBio does is rather than a bunch of institutions or a bunch of customers having to do all those steps individually, we really take the data down that refinement path and moving to a product that can then be analyzed much more quickly and much more readily, and then ultimately matched up with other data sets. So again, your prior question, we draw that primarily from Vanderbilt, but certainly on our radar to think about how do we move some of that refining to other data sources outside of Vanderbilt.
What's the range of data, and what's the range of things beyond data, things like biosamples that you've been able to gather? And how unique a collection is this?
Yeah, it's I guess I often describe it in concentric circles, and so the biggest concentric circle is all of the electronic health record data. We have about 4 million subjects of worth of electronic health record data as kind of that biggest concentric circle. That's everybody that's interacted with Vanderbilt in some fashion over the past 25, 30 years time frame. I think one of the unique aspects of that is that window of ascertainment. Vanderbilt was one of the early adopters of electronic health records. They started bringing together the pieces for electronic health record assembly in I think 1999, 2000 timeframe. So there's like 25 years of history at this point, and there's a lot of paper records that were ported over as well. So that longitudinality of information over that such a long period of time is an important piece of it. We have biospecimens right now in the form of germline, primarily germline DNA. So those are the discarded or residual blood samples collected secondary to clinical care. Again, somebody consents to participate in the biobank. We wait till they have a blood draw somewhere in the health system and then get access to that residual material. So about 350,000 samples sitting in the freezer in that regard. And then over time, we've worked on various initiatives to turn that into useful data. And again, going back to my resource analogy, raw biosamples are great to have and certainly better to have collected than not collected, but it's only a step in the analytic workflow. Ultimately, you need to turn that into genomic data to be able to subsequently do the experiments. So we have about 250,000 old genomes, and I suspect we'll talk a little bit about the AGD initiative, how those came about. But that's a big genomic data set we have access to. So some of the other unique aspects I highlighted longitudinality. I think certainly the depth of the clinical information is a unique differentiator here. Having access to all that unstructured information, things like the notes and the imaging and all the other information that gets wrapped around the healthcare data, that is a definitely a unique aspect of it. The fact that we span all different disease areas as opposed to certain specific disease areas, that's another differentiating characteristic. Age diversity, site of care diversity, ancestral diversity are all important pieces that feed into the data as well. And those are largely a factor just where Vanderbilt sits in the healthcare ecosystem. It's pulling from a big catchment area. They have a wide range of clinics from primary all the way up to tertiary care. And then just Nashville and the surrounding Tennessee area do have a great dose of different ancestral diversities that feed into the data set.
What's the range of things you've seen clients use the data for? And given its long history, is it possible to do a retrospective natural history of a disease with this?
Yeah, it really is a robust and rich workbench in that regard. And I would posit there's no limit to the questions that can ultimately be answered with this type of resource. That was a big motivator for Vanderbilt in terms of thinking about its mission. That there's so much science and only so many scientists at a place like Vanderbilt. So opening this up to being able to pull in industry partners. Um, we try and work all across the R&D chain or pipeline or however we want to describe it, thinking primarily in the pharmaceutical space, but certainly applies to other parts of life sciences as well. So whether it's early research and discovery activities, trying to characterize disease populations and ultimately help discover the new drug targets that a biopharma company may develop into a subsequent drug. That's primarily there. That'd be the biggest use case, and certainly that fusion of the genomics with the clinical phenotype from the electronic health record, building very very specific or very exquisite populations to do those types of analyses because we have all that depth of information. You can start to simulate clinical trial endpoints in that regard as you're designing those genetic studies. You can start to look at more complex phenotypes things like disease progression, drug response, all of those things are possible with the depth of the clinical information we have access to. Once a target's discovered, certainly target characterization becomes another step. So understanding, are there other phenotypes that may be implicated or affected by modulating that drug target as something we move into the clinic. Really trying to understand, are there potential safety signals that could come about. Myself and my colleagues at Vanderbilt have published a couple different papers on using these techniques to assess safety profiles in that regard. As you move into clinical development and clinical trial design, certainly using it for clinical protocol development. We've done some work in what are called synthetic control arms, so taking a clinical trial protocol and identifying real-world patients that would match those characteristics and really trying to understand what that baseline standard of care is. And it's particularly useful if you're running a single-arm study. You can fill in kind of the comparator arm using real-world data in that regard. And as you move even further down the pipeline, thinking about drugs and treatments that are out in the market, really studying the real-world uses of these therapeutics, thinking of things like comparative effectiveness, the launch of a new drug, trying to understand how does that alter treatment paradigms, what are the health economic implications of that, does it reduce or increase the utilization of care? These are all areas that we've supported over the years. Things like natural history of disease. How does somebody move through the healthcare system? How does that disease progress over time? Whether it's a diagnostic odyssey or trying to understand what's the process to get somebody diagnosed and ultimately somebody treated. Again, these are various flavors, and just depending where the question's coming from, we'll emphasize different pieces of that patient journey. We'll draw on different parts of the record. But again, going back to my opening on this one, it really is a rich workbench, and it comes up with asking, or it depends on asking intelligent questions, but we haven't yet hit a kind of set of questions that we can't answer in this domain.
You alluded to the AGD. This is the Alliance for Genomic Discovery. This is a collaboration that you've done with the sequencing giant Illumina and group of pharmaceutical partners. I'm wondering if you can explain exactly what that is.
Yeah. This was an initiative to kind of really turbocharge a few different things. Certainly the genetic data and resources that are available inside BioView and bioextension within NashBio, but then ultimately to support a much broader scale of drug discovery and some of the use cases I just walked through a moment ago. The goal in simple terms was to move away from kind of sequencing or analyzing small cohorts of subjects within the biobank. So historically, we and my academic colleagues at Vanderbilt would go and design a cohort and then withdraw a specific patient population from the biobank and then charter the sequencing of that data set. So maybe 1,000 subjects with kidney disease or 3,000 subjects with rheumatoid arthritis or something like that, which is good for the specific study, although there's time lag and certainly cost involved in going and getting those samples out of the biobank and turning them into useful data. And then the reusability of that data is somewhat limited. You end up with a real patchwork of data when you take that approach, and it's hard to combine those things without introducing batch effects and various confounds into the data set. So the AGD was a goal of, it came up with a goal of basically, can we sequence all or at least a significant portion of the biobank in one swoop, where you're able to deploy the same protocol, the same analytic approach across all of it. You'll have a much more uniform data set that can then be used in that workbench analysis or kind of platform or workbench analysis or analogy that you can use over and over again in repeatable fashion. So it took us a couple of years to figure out a path. And again, another example where there's some stops and starts along the way. We really started in early 2020, just before the pandemic, in terms of trying to figure out how to accomplish this goal. We did have the benefit of a couple of these initiatives having gone on before us. The UK Biobank in particular had organized a couple of consortium initiatives to accomplish the same goal with their data sets. And so we had a bit of a template laid down in front of us. But anytime you're trying to do an initiative of this scale, it's still you have to put it in your own context and figure out your own path. And so, as you mentioned, we got partnered up with Illumina, and then through a joint business development effort, we're able to recruit eight large pharma partners together to ultimately fund the sequencing of that data set. And the premise for them was to help us generate the data. You'll be able to use it in your own research and development activities and kind of all those different things we talked about a moment ago. And then the benefit for us is that we were able to help provide it back to our colleagues at Vanderbilt, so to power the research enterprise and the research infrastructure at Vanderbilt. and then through NashBio being able to reuse that data after a period of exclusivity where the members have exclusive use of it. We're able to take it into, as I describe, our retail channels where we can then cut it up and use it in smaller bits and pieces with other companies.
And is there an ongoing relationship with these pharmaceutical partners?
Yeah, it's so it took us 2 years to generate the data. We started in January 2023. We just announced the completion of the first phase of sequencing in March. 250,000 whole genomes under that construct. So really a tremendous research resource for all of the participants, and on time and or actually slightly ahead of schedule and on budget, which for anybody who follows these types of initiatives knows that those two adjectives are relatively unique in this type of space. Usually, it takes longer and costs more money than what's initially estimated. So we were quite pleased with that. We are actively engaged with the different members of the alliance to figure out ways to help them continue to use these data. So it's as I highlighted, kind of the depth of that clinical phenotype. There's lots of complexities in terms of figuring out how to digest and make use of that, and so we're working with a number of them in that regard. There's a number of potential add-on enhancements to the data set. So some of the things that we've been working on would be extending it into specific therapeutic areas and making example, maybe making use of some of the unstructured data that we have access to, transforming that into an enhancement focused in cardiology or hepatology or certain therapeutic areas. And so working with a couple different members to build out those types of enhancements that then they would be able to use in their own activity. And then as a group, we're collectively looking at ways to extend the alliance. So given the success we had with the first version, looking at active ways to extend that, whether it's more additional sequencing of DNA samples. Also actively discussing proteomics is another omic layer. That's one of the big trends we're seeing in this space, is the move to more multi-omics or multi-omic analyses. And again, I'll cite the UK Biobank again. They're really a leader in this space where they've opened up a proteomic layer, and so it's a natural area of interest here at NashBio and Vanderbilt. But also some potential in areas where we would be unique. And so like thinking of other biospecimens that we might be able to tap, there's a lot of interest in transcriptomics and biomarker discovery, which would be able to use other biospecimens beyond the residual blood samples. Metabolomics is another area where we're starting to see more and more interest. And so actively discussing all these areas and trying to figure out amongst the group of members which would have the most interest and support from them as they're thinking about driving their own R&D program.
It's compelling to think you could marry the genomic with all the phenotypic data. I'm wondering, though, have you done anything to address concerns about a lack of diversity in genetic data, which has been endemic to the entire field?
Yeah, it's the history of a lot of these data sets is a heavy emphasis or heavy focus on individuals from European and even Northern European ancestry. That's where some of the initial biobanks were initially developed, whether that was actually in Europe or here in the United States. But there does tend to be there has been an enhancement or a focus on those populations. So one of the things we did with the AGD initiative was intentionally tried to enrich for subjects with non-European ancestry, more diverse ancestral backgrounds. So the first 35,000 subjects that we sequenced in the AGD were individuals exactly of that background, and that was specifically to try and start addressing some of the gaps in diversity of genetic data. So all of the members got access to that data set right from the get-go, and again, something that came back to the Vanderbilt research community in very short fashion. And I know it's been an active area of study both at the Alliance members and at Vanderbilt in terms of making use of that data. It's, as far as we're aware, it's one of the largest collections of diverse ancestral whole genomes at this point. We know there's other initiatives underway that'll surpass that count. But for the time being, it's an incredible resource just to start to study some of the disease areas that maybe are less well represented in European populations, or say more enriched in non-European populations. Trying to understand the specific context of how genes and disease fit together. Even just understanding if are there differences in different parts of the genomic code across some of the major ancestries. So we were quite pleased that we were able to put that together as a starting point in the alliance. And we're waiting to see what's going to come out of it. I know my colleagues at Vanderbilt are working on a publication that'll kind of highlight all the different uses of that first diverse ancestral cohort, and look for that sometime later this year, highlighting some of the initial findings that have come from it.
And what's the business model? Is this a subscription service? Are you engaged to do work around specific projects? Is it something else?
Yeah, it's so again, my referring back to my background, coming professional services and consulting background, we really did start with a services first model here at NashBio. And practically, what that means is that somebody would come to us with a question of some sort in their R&D needs, and we would our one of our teams would work together with the client to ultimately develop a data set and then potentially analyze it um, to advance that question. So again, kind of a people first or services first model. It would typically have been our hands on the keys, certainly on the source data, our hands on the keys, and then maybe the client doing the subsequent analysis once we've built out the data set. It does certainly There's certainly value in having expertise working through this data, both real world data and our specific flavor here at Vanderbilt. There's all kinds of nuances that come about from the practice of clinical care and the fact that this data is derived for other purposes and being reused for some of these applications. And if you aren't familiar or fluent with some of those limitations, you can get yourself into funny cul-de-sacs pretty quickly. So we do see a real value in that services component. That said, it's not universal, and we've definitely seen a shift over, I'd say, the past 5 years horizon, where there's more and more capability and competence being built up in the biopharma space for interacting with these types of data, and not necessarily wanting to work through a services fashion. So we are, in parallel to our services model, in the process of standing up a new model that is going to be more technology led, where we'll be able to provide direct access to the data sets through what we're calling our Totem TRE. TRE is kind of the term of art that's grown up in this space, trusted research environment. It basically means a secure workspace sitting somewhere in the cloud. Totem, in our case, is the name we've applied to it. Totem is Latin for whole, in this case, access to the whole cohort, being able to assemble the whole picture of the patient. And so in this new model that we're just standing up right now, we'll be able to welcome clients into that TRE environment. Those who know what to do with the data will be able to interact and engage with it directly and analyze it subsequently. Our services teams, whether it's clinical or data science focus, will still be available to help, and that's where we think it becomes quite a complement. That going back to some of that expertise that maybe somebody doesn't have, they have the data science capability, but they lack the clinical context of how did this patient actually flow through the health system and what were the providers doing at that point. So we'd be able to tap one of our clinical teams. So we're quite excited about the new launch. We had a beta program running for the past 6 months and kind of an early homegrown version of the offering. We're just going through stand up right now and looking to start to welcome some clients into this new Totem model over the next probably month to 6 weeks time frame as we move that into beta testing.
So, what would you say having this resource available allows drug companies to do that they might not be able to do otherwise, or that they can do more efficiently and cheaper and faster?
It's a resource efficiency question. I think that really is the key value provider, value driver here. And what I mean by that is that this data is all collected, already previously collected. And so it allows for kind of very rapid hypothesis generation, testing, and analysis that isn't necessarily possible in kind of a primary collection setting. And I'll give an example of that. We did a project a couple of years ago with a pharmaceutical company that wanted to understand the progression of liver disease, and they wanted to look at individuals with kind of similar risk factors, diabetes status, obesity, several other complications, and kind of looking over a 10-year history to try and understand what happened before the diagnosis of disease, and then studying what happens after that diagnosis. So you think of like a 10-year history, somebody trying to do that study, if they were to set up a registry of some sort, picking people who for 10 years are going to follow and then hope that they manifest the condition, and then subsequently study them just as an early research study, that's a really challenging thing to justify from a cost standpoint. But the fact that we're able to derive that information off of the health record, we have that 10-year history for a large number of patients. We have all of those procedures and kind of documentation that comes about, and then we can, once we've sorted patients into disease, not disease, or fast responder, not responder, we can then look at what happens subsequently. So you can design really elegant studies using all of that information. And then in this case, it was a genetic study, so they wanted to compare the DNA code from the two different patient populations. They were using it to try and identify drug target hypotheses. And so it's a combination of that too, being able to get access to that effectively instantaneously. Maybe not. Maybe there's some transaction time or there's some familiarity time, but being able to design a study like that and get access to it in the course of weeks or months is far more efficient than trying to set that up and to collect that primarily. If you had to do it from a primary standpoint, I think you'd be hard pressed, or you would hardly see any types of studies like that.
One of the striking things about your website is you don't see the term AI showing up anywhere on it. And I'm wondering to what extent, if any, you're using AI in integrating into the product offering.
Yeah, it's not intentional that it's not up on the website, but I mean, we're not riding the wave that a number of other technology companies are, where AI everything everywhere all the time. We are actively using AI and related tools in a number of different ways within NashBio. Certainly, our engineering team has been a big adopter of that, and just being able to accomplish more things more quickly through the use of coding copilots and support like that. We are active users of things like natural language processing and LLMs in terms of interacting, curating, extracting information from particularly some of those unstructured resources that we have access to. Thinking about a giant corpus of a billion clinical notes, obviously a large language model is well positioned to be able to engage with that, and so we're actively deploying tools such as that to really get information out of those notes, help with synthesis, help with feature extraction, to get the information content out and really find the useful from the less useful. Um, we've done through discussions with customers directly, particularly as a data provider. Some of the advice we were given is to really focus on some of those uses. So like things like cleaning and normalization and quality control. And so like deploying AI to really make a better data product. There certainly are downstream analytics that I think we're all trying to figure out is AI really a tool to help integrate all the clinical phenotype and all those multi-omic layers and kind of see things that the human mind is not able to see? And I think the answer is yes, but it's still early in that phase. I don't think we quite know yet what the LLM equivalent is in that biological context. So, what is the model that's going to tie all those things together and say, all right, yes, here's the drug target that comes out from all of that. And so, rather than spending a bunch of time trying to in kind of more speculative areas, we've chosen to focus on some of the stuff higher upstream that we know customers would have to do themselves, and we'll let some of those analytic areas play out a bit longer, and then figure out where we want to go.
You've talked a bit about your relationship with the pharmaceutical industry and the way that drug developers might use this data, but I would imagine it has great value on the clinical side in terms of supporting treatment decisions and the like. Are providers trying to access this data in any way?
It's certainly yes, as another valid set of use cases. A lot of that does tend to sit on with my academic colleagues at Vanderbilt. So that's a lot of the primary use of the data set, maybe identifying undiagnosed disease populations and trying to figure out how you could short circuit that diagnostic journey that maybe a patient went through. There'll actually be some interesting findings about that coming out in that paper I mentioned from AGD, of like undiagnosed disease even with signals, and so therefore can we inform care decisions much sooner than that. We as an entity, and whether this is my own bias coming from having worked in the pharmaceutical space before, or kind of more obvious use cases, that's the direction we gravitated to. There's not a maybe a slightly less obvious client for some of the things you had mentioned. That certainly, as I said, Vanderbilt's researchers are certainly making use of it and pulling it back into their own domain. But we haven't. We'll certainly work with somebody that wants to work in that space, but we haven't done a lot of that specific type of activity in the commercial realm. So certainly possible, happens all the time. A large corpus of publications coming out of Vanderbilt that highlight those things, but it hasn't been a large focus for NashBio. We're working more directly in kind of the medical products and drug space, and then ultimately driving benefit to the patient population by helping bring those new products to market somewhere down.
Ago, the work you're doing is something we might have expected from the National Institutes of Health. That's a reflection of technological changes, but given your ties to an academic medical center and working in an industry that has significantly benefited from public investment in research, what are you seeing with regards to cuts to NIH and other federal investments in scientific research? And how big a concern is this at Vanderbilt or with your pharmaceutical partners?
I think I'll divide it into kind of different phases, but I guess as a macro comment, I think it's something we should all be concerned about. I mean, the NIH has a long history of funding biomedical innovation. They're one of the largest funding sources in the world for this type of early research, and has powered countless discoveries over time, often without necessarily knowing what was going to come out of it. That it's one of the beauties of NIH research is kind of the serendipity. Something gets funded and it leads to something else, to something else, to something else that ultimately provides a new drug or therapeutic. So I think it's something we all have to be concerned about. That is, as that money is throttled back, what's going to happen to the innovation ecosystem? There's obviously a huge biotech presence here in the United States. It's one of the crown jewels of our economy, and so are we cutting out the legs or the seed corn of future innovation years down the road with some of the actions that are occurring right now? You mentioned, yes, certainly with our ties to Vanderbilt, and a lot of the major academic medical centers, academic institutions are feeling the both just the uncertainty as well as a number of the outright cuts that have been made to NIH spending, and taking action to adjust their own cost profiles, maybe shutting down certain branches of research or having to make decisions that are detrimental. I'd call that sort of indirect impact to NashBio. So if Vanderbilt ends up making changes to some of the research infrastructure they have, some of that may flow through to us. Something may not be available quite as quickly. They're not able to refresh data as often, or maybe they're not able to collect as many samples into the biobank as they had. So that'll slow down the kind of the rate at which we can make use of it. Maybe the more direct impact to us has been less obvious yet, but that's where, as kind of that layer of innovation gets suppressed, I think that will start to have impact in the pharmaceutical space, whether it's biotech or pharma somewhere down the road. That we're just going to see fewer drugs coming to market. We're going to see fewer diagnostics and fewer treatments being developed, and that's as a few years down the road. Unfortunately, we're not going to know exactly what that is, but that's definitely something I'm worried about, given the sources of funding and how this whole innovation ecosystem has worked for the past 50 plus years.
It's been a tough environment to raise capital, but as far as I can tell, NashBio hasn't had to go the route of conventional venture investment. How are you funded to date, and is there a plan to raise additional capital here?
Yeah, it's that is certainly one of the benefits of Vanderbilt having invested so much in these resources prior to the creation of NashBio. I didn't have to go build the biobank. I didn't have to go build a lot of the integrations into the electronic health record. So we were able to start from a pretty advanced state in that regard. Vanderbilt did seed capitalize NashBio when we first launched the company, so we had kind of working capital, initial growth funds coming in into the business right from the outset. By nature of the work we were doing, we were revenue generating right from the start. So unlike a biotech in the drug discovery space, where you're burning cash for 2, 3, 5, 7 years as you're working an idea from discovery through to clinical trial, we were able to generate revenue from the outset of the company, and then able to get profitable fairly early in our history as well. I think by year 3, we were in a profitable stance, and have been so since then. That said, there's certainly a lot of things that we could do and go more quickly, and that's maybe the limitation of the model we've had. Haven't had to raise money, but we have gone more slowly in certain areas, and you could say positively that's built a discipline into the team. We haven't been caught by the latest fad because we have the resources to go pursue it. We've had to be more balanced in that regard and make sure there's demand for whatever it is we're going to develop. I do think some of the longer term things we'd like to accomplish, and I'm thinking particularly of like opening up other data sets, or opening up other data sources, I should say, the large scale generation of other omics data. Those types of activities are going to require more funding than what we can generate from our own operations. Certainly, replicating the AGD model, where we're striking up partnerships with industry and leveraging a form of non-dilutive funding in that regard. Absolutely exploring that, and constantly discussing with Vanderbilt as well. Like, is there a trigger or a point at which we would go seek more traditional venture or growth capital at some point? So, not sure where that path's going to lead. It's certainly on our minds, and I think to really accomplish some of the bigger goals we would like to with NashBio, we'll have to get there at some point. But we've been quite successful to date, and it's we'll continue on with what's worked for as long as we can.
As you think about the opportunities for using the data you've gathered, what's the plan for growing your offerings or expanding the data sets or the markets you serve?
As I said, the biggest expansion at the moment is the kind of the addition of the whole new business model, kind of the totem TRE model, in terms of being able to service customers in a fundamentally different fashion. Certainly wanting to make as much as possible out of the data that we get from Vanderbilt. And so what I mean by that is like identifying maybe data features or data sets that are already collected at Vanderbilt but aren't connected to the main health record at this point. And so working on being able to locate and de-identify those resources. Imaging would be a good example. Radiologic imaging would be a good example of that. That wasn't traditionally in any of the data Vanderbilt de-identified. So over time, we built up different pipelines for de-identifying what are called DICOM files. That's the raw data, the way medical radiologic imaging is generated and stored in a health system. And so looking to do that in other opportunities. We know there's certain therapeutic areas that tend to sit in siloed data sets that aren't connected to the main biobank. And so absolutely looking at ways to be able to get access to those and then build pipelines. One of the big pushes, and this goes back to the AI LLM focus, being able to extract more information from all those data types. We've definitely had that feedback from clients before that some of those unstructured data types are sexy sounding or exciting to use, but they're just frankly hard to work with. And if you don't have the data science capability sitting there staring at a billion notes or a million MRIs or something like that, you're really interested in the information content. Unless you're training models directly yourself, you're really interested in the information content. So one of our big pushes at this point is to deploy some of those tools in service or in kind of pre-processing of these data sets and really getting that information content out so that going back to that point about speed and kind of rapidly deploying these data, we'll have those things ready to go. Working on a therapeutic area alignment at the moment, working through different therapeutic areas where we're seeing the most demand. But again, really trying to make that complex data easy to use. And then new omics layers that we've talked about as well, moving down into proteomics, metabolomics, transcriptomics. We're excited about all those and trying to figure out which order we move through those layers, which actually have value to the customers we work with at this point versus just generating data for the sake of generating data. We obviously want to make sure that the data we generate has utility for our clients. And then probably fourth horizon beyond that would be expanding beyond Vanderbilt. How do we replicate this model, drawing on other data sources, whether it's here in the U.S. or outside the U.S.? We've had feedback from our customers in a number of different fashions that you guys could help us collect and munge data together to make it all interoperable from other sources, really around the world. We know there's opportunity there, and we're trying to figure out how to do that. And again, in kind of measured fashion, given the resources and bandwidth we have, but it's definitely on the radar.
Leeland Ekstrom, co-founder and CEO of NashBio. Leeland, thanks so much for your time today.
Great. Thank you for the conversation, Daniel.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.